notes. Okay, thank you. Before starting, we wanna ask two things. So please, Zoom attendees, please make sure you are muted during the lectures. And then if you have questions, please hold your questions until Q&A session. But if you are attending via Zoom, you can type your questions anytime in the chat. This is today's agenda. So I'm going to briefly talk about what is freeze fracture replica immunogold labeling as a basic background. And then I'm very happy to have Dr. John Rush, my former supervisor, to give his special lecture about the brief history and the basics of freeze fracture replica and replica labeling. Then following by Debbie and Conan's talk, they will give you more practical sessions. So when I started planning this workshop, our target audience was our students or trainees who are not familiar with freeze fracture EM or even EM itself. But when we started announcing this workshop, we were a bit surprised and then super happy to know many people are interested in this topic. And then at the end, we got more than 150 people registered. And then again, another happy thing for us is about more than 2/3 of attendees are outside of Jupiter campus. It's unexpected, but we are so happy. And then as you see, there are so many people from all over the world and then more than 10 countries. So I have to say good morning and good afternoon and then good evening to everybody. Thank you again for attending. Okay, let's get started. So my talk is about the brief overview. What is freeze fracture replica immunogold labeling? And what can we do with it? Many people in this workshop are not familiar with this technology, so let me back up. What do you imagine when you hear freeze fracture EM? I can imagine, no, sorry, freeze fracture EM. So I can imagine you just simply imagine something like this as a regular EM image, something sectioned, and then we can see inside of the cell. But what about this? This is the same region of the brain, and then shown here in the same magnification as you see scale bar on the bottom. So this is a part of the brain stem of the mouse. And then actually we are seeing the same structure here, like axon bundles in both sides. And then we have cell soma in the middle. But these are totally different looking. But you can imagine similar impression of the structure. And then if we focus on this boxed area, what we are going to see. Left side is pretty familiar image maybe with you. We have a beautiful synaptic vesicles filling in one presynaptic terminal. And then mitochondria in the terminal, and then this terminal is contacting onto cell soma. But on the right side, this is freeze fracture image. We can still see synaptic vesicles in a cross fracture of a process next to the cell soma. But this cell soma looks totally different, much flatter. Why? Because this is membrane surface. So let me use one analogy here. You can imagine watermelon. So when we cut a watermelon, we can start seeing red part inside. So this red part section image is like conventional TEM on the left. And then freeze fracture image can show the surface of the watermelon, green and black band. So let's say these black bands are tight junction. What is tight junction? Tight junction is one of the adhesion molecules and then locating on the cell surface and then making this beautiful strand structure. This picture is from one review article. And then as you see here, tight junction in a thin section image at the contacting place of two cells. We have these contacting points. But now you can imagine it may be super hard or almost impossible to reconstruct or visualize this strand structure from this type of image. So this is a big advantage of freeze fracture EM. We can see membrane structure in 2D view. Then you may ask me, so why is freeze fracture EM useful in neuroscience? Because all brain functions are based on cell-to-cell communication, and many key protein molecules such as ion channel, transporters, and receptors are all on membrane. And then this theory is also true for organelles in the cell. When organelles has some interaction with cytoplasm or other organelles, again, membrane is a contacting part. So it's why we want to analyze these membrane proteins. Then what do we see on freeze fracture replicas? What we can do for our research? These are our next question. It is why I organized this workshop, and then we hope you are going to find some hint or answer from this workshop. Okay, so this image became more familiar with you from our flyers. This is a part of freeze fracture replica immunogold labeling. And let me back up the watermelon theory again. So here, if I put same color, we have red part inside of cell at the edge. And then rest of part are green part. So in the membrane part, we are actually seeing two different views of the cell membrane. One is protoplasmic phase, P phase, and then another one is extraplasmic phase, E phase. And you can see lots of bumps here. These are integral or intramembrane proteins, IMPs. They are sometimes sparsely localizing on the membrane, or sometimes they are making groups or aggregation. But actually, these colors are added by Photoshop. So what we are seeing in the EM is something like this, all black-white. But using black-white image, you may see better IMPs. These are sparsely localizing, and then these are making a group. And then you also see gold particles better. On this P phase, we have small gold particle and then large gold particles. So for this case, we used this small gold particle as a cell type, cell marker labeling for GFP, just like a green fluorescence protein under the fluorescence microscope. Because we only have black-white, we have to use gold particle, and then we see specific cell type by this result of labeling. And then these larger gold particle shows a kind of one of the calcium channel here. So this is an image. And then what we can do using this technology is we can analyze membrane protein distribution, and then we can now discuss the function of the proteins. So this is just a brief overview from my side. But if you have a particular question, please ask right now. We know there are many new people to this technology, so you may want to confirm anything else, or do I have any chat questions? Nothing? Okay. Okay. So maybe I spoke too fast, but anyway, if we don't have any questions.
There's one question.
Okay.
How can you tell the difference between P phase and E phase?
Oh, that's a great question. Thank you for asking. So let's see. Here, if you target these IMPs, this E phase has, maybe this way, has less IMPs. And then this P phase, because of this gold particle, it may be hard to see, but P phase usually has, not usually, always have more IMPs. It is because of just a protein molecule preference, which E phase, P phase, they wanna go. And then lots of protein prefer to go to P phase. That's why, generally saying, don't wanna, okay.
If I can step in a moment, Naomi-san.
Sure. Go ahead.
So the easiest definition to remember is that the P phase or protoplasmic leaflet immediately behind it, as is seen in the middle of the screen. Do you see my arrow now?
Nope.
Okay,
Yeah.
so
Yes.
in the middle of the screen, right immediately behind that membrane is the cytoplasm, the protoplasm of that cell. Whereas the upper portion, the extraplasmic leaflet, immediately behind that upper membrane, or the one in the lower right, is the extracellular space. So that is the extracellular or extraplasmic leaflet, and that's the E phase. And in most cases, but not all, in most cases, there are fewer particles, IMPs, in the extraplasmic leaflet to the outside, and far more of them cleave to the protoplasmic leaflet, stay with that for unknown reasons. We think it's because of cytoplasmic proteins maybe helping to bind them in the cell membrane, but we really don't have proof of why this differential cleaving occurs.
Thank you, John. And I got another question. Do you have watermelon example of E phase and P phase? Sorry, I don't. But yes, let me explain a bit more. So we have membranes as lipid bilayers. And then when we fracture sample, biological sample, this lipid bilayer split. Then we are seeing outside of membrane half leaflet of lipid bilayer from the inside of room, and then this is like a ceiling. And then P phase is protoplasmic phase, and then we are looking down protoplasmic phase. So yeah, can you imagine how I can make this watermelon cartoon? Yeah, because we have to split the green part of skin into middle, right? Yes, so Jim, it's too hard. Okay, so another question came. Can you see where docked synaptic vesicle would be in this photo? Actually, we sometimes can see docked vesicle just at the edge of this. So this is a red part of watermelon, cytoplasm. And then this is like an edge of cytoplasm to membrane. So by chance, we can have this omega shape of vesicle right here. And then morphologists, or we believe these things are actually remnant of the docked vesicle after fused.
Naomi-san,
Yes.
in a review that I wrote about 30 years ago, it's the authors, you can look it up. It's Rash, Walrond, and Morita. There are images primarily from John Walrond, but a few from me, of docked vesicles at motor nerve terminals in the neuromuscular junction.
Yes.
And the corresponding arrays of what we now know to be calcium channels as the active zones on the surface. But you have to go back about 30 years. I think that was 1987.
Yes. Also, one of the famous picture from another John Heuser.
Yes.
He did frog neuromuscular junction with stimulation. Actually, we can see the synaptic vesicle fusion as a membrane structure. So that's interesting thing.
And I think John Heuser gave us images of his to put in that same review, but I just don't remember all the images now.
Okay, and then another question came. What happens to the cytoskeleton anchored to the membrane? Is it lost? And what are the requirements in terms of antibodies you can use for immunogold labeling? So cytoskeletons are there. So, but we have to use additional trick of this freeze fracture. After fracturing, if we evaporate water good enough by rising up temperature a little bit, not melting. So ice is evaporating, and then we can start seeing the deeper portion of fractured membrane. So it's so-called deep etching technology. By doing that, we can see the cytoskeleton just below the membrane. But if it's in a cytoplasmic portion, it's gone by washed out. And the following question is, what are the requirements in terms of antibodies you can use for immunogold labeling? So yes, this is a great question, thank you. So this is still immunolabeling. So you may know from hard way, Western blotting or histology or immunofluorescence, some antibody worked for your project, but some antibody didn't. So this is still true for this freeze fracture immunogold labeling, but we are using SDS that Debbie is going through, but SDS is used for Western blotting for denaturing protein. So this technology is higher sensitivity because of that process. So by denaturing this 3D structure of protein, it's easier antibody to attach to the epitope. So with our experience or other people's experience, finding antibody is a little bit easier than sometimes the fluorescence. But also, as I explaining, E phase, P phase, some protein has preference to go to P, sometimes go to E. Then, again, David's talk will show that, but we have to select which side of epitope is recognized by particular protein in terms of, like in the transmembrane protein, we sometimes have N-terminus goes to external, C-terminus goes to internal or external. So epitope position, either external or internal, are important. If it's opposite, yes.
If after I finish my talk, we have a world's expert here in Jim Nagy, who's worked for many years with us to design the antibodies that work best for the particles in freeze fracture. And I think we should call on him to give a minute or two or several minutes explanation of how the antibodies are designed, et cetera.
Yeah, that's a good idea. When we have time, let's go through that. Okay, so now, okay, another final question. What determines whether a membrane protein cleaves on the E phase or P phase? So this is a general question and then frequently asked question, I think. I think correct answer is nobody knows. Is that correct, John? So, but we can imagine maybe transmembrane protein sometimes have a bigger external tails, or some adhesion molecules are really attaching to the external. Or some protein has long C terminus internally and then bound to the other scaffolding protein, for example. So that balance probably makes the preference of individual protein which they want to go. But sometimes it's not true. This protein has a very tiny external loop, but still goes to E phase. So when we analyze new protein, to be honest, we don't know this protein goes to P or E. So we have to test the different antibodies having different epitope to external or to internal.
And the important concept here, about 40 years ago, Birgit Satir defined the particle partitioning coefficient. And it's been found and repeated many, many times that any particular class of membrane proteins almost always has a particle partition coefficient of 100% to either the E or the P, that they will all go to one side or all go to the other. Only a very few more or less cleave evenly, that is 50-50 to both membrane faces. So it's a good marker that we can make use of in the identification, the structural identification. And once they're labeled, you can know that if you see something similar, but it's on the wrong face, it's almost certainly a different protein.
Okay, thank you. Okay, so with that, I think it's almost time. We should move on. So again, I'm very happy to have John Rush, Professor John Rush, in this workshop. So he is my former supervisor in Colorado State. And this is his brief bio sketch. But rather than reading this, I want to introduce in my words. I joined John's lab 2003 and then worked about 4 years in the lab. I had very limited experience with freeze fracture EM when I joined, but I was given some replica samples from the day one. And then I went to EM, I started examining replica samples literally every day, and then from the morning to the night, and really every day, all day. Then John usually comes to the EM room around 4 p.m. He's saying, "Naomi-san, having fun." And then he joined me, and then we still kept imaging until night, sometimes close to midnight, not sometimes, often till midnight. And I was doing that, and then John kept speaking about freeze fracture or replica or biology while we are examining replica together. So I learned all the basics, all the principles of freeze fracture EM from John Rush. So I want to share just one story, although I have a bunch of good memory. After about 6 months joining John Rush's lab, John took me to a day trip, so-called Vedavu. It's a native Indian sacred place. And it's so beautiful, as you see, big rock formations. But after 6 months in a training, what I saw there, and then I shouted was, "Oh my gosh, look, it's a soma E phase." Or, oh, look, it's a myelin cross fracture. Yes, this is really true story, how I was trained by Dr. Rush, and then how people can get into the morphology. So before going too far, I wanna introduce Dr. John Rush for his talk. So please join me to welcome John Rush. So now would you share Your screen, John? Stop share.
So I'm not quite sure how this screen works. I wish to use, if you can see this PowerPoint present. Oh, I have to do share. And so just give you, I've recently moved to Texas, which I may be leaving in the next year or two, but for the last 40 years, I've been working at Colorado State University. I retired a couple of 3 years ago. My talk today is going to be, I'm going to start the first 20% of it, some of the original goals of freeze fracture and freeze etch, just so that everyone can have a basic understanding of what people were working on. It has a 70-year history now, started in 1957 by Russell Steer, who introduced all of the major elements of how to do freeze fracture, and still has some of those early images are still the highest, among the highest resolution of all freeze fracture. So we owe him a great debt of gratitude, but we can't talk about his work today. There's not enough time. The second part of the talk will be the development of an application of both SDSFRL, and you will get a lot of abbreviations, which has an earlier antecedent called frill, and we've revived that simpler name because our version would have had to have been called SDSFRL GMFF, and that gets way too complicated, and we'll show you what that's about shortly. I lived in this area of Colorado. This is a few miles from my house. I lived at 6,800 feet elevation. I now live at 20 feet elevation in Texas, literally. And Nami-san, I've got material, a bar. I can move the bar. It's keeping me from seeing my own screen. Okay, so the first part of this talk will be about how the replica is made, just briefly. And if I can back up so it doesn't skip too fast. So in the first 40 years, the main questions were, how do you make a freeze fracture replica? How do you clean it? How do you examine it? What is the limit of resolution, et cetera? So the basic technique is that you have to have a very cold frozen sample, so cold that it is brittle. And when you apply a shearing force to it, whether that's a dull instrument like a very sharp razor blade, or you just break it in half as if you took a stick and broke it with your hands, you want a brittle fracture. And that's done under a very high vacuum. And once immediately upon breaking it, in most cases for freeze fracture, you have to deposit something that will be visible in the electron microscope. We use an electron dense, insoluble inert metal. And the primary ones that have been used are platinum, chromium, tungsten, tantalum, but a few others have been tried. That thin film is put on at an angle, and then we put a backing film on that, a support film of ultra thin carbon. And that in this old image, this is about 40 years old. The specimen can be fixed or unfixed. You cannot use glutaraldehyde for frill. You can use glutaraldehyde for regular freeze fracture, and I'll explain that later. But it's usually put on a gold specimen support of some sort, but it can be copper or other kinds of hats. That sample is frozen some way. There are many different ways to freeze. This is just an early illustration using liquid freon as a cryogen to rapidly quench and lower the temperature and freeze it more or less vitrified. It can be cryoprotected with an agent like glycerol or just left as is. It can be frozen with methods like high pressure freezing or slam freezing against a liquid helium cooled copper block. There are a number of different ways to freeze the sample. After the sample is frozen, it is placed in a vacuum chamber. And this is the old Balzer's 301 for those of you that recognize. And under high vacuum, that's when the fracturing occurs. this fracturing stage is here. You don't need to have the details at the moment. And then immediately upon fracture, it's shadowed with the electron dense material. We typically use platinum. And then immediately after it's shadowed with platinum, it's then this is an old fashioned carbon arc where you deposit a few nanometers, 20 or so nanometers of carbon on top of that to make an ultra thin film. So this step here is the fracturing step. Again, the dull instrument here is an ultra sharp, it can be a diamond knife, glass knife, or whatever, but at the molecular level, that's a dull instrument. It starts a fracture plane that goes through the tissue and separates at the lines of least resistance, which are usually within the membrane bilayer. Normally, if I can move this, normally we go, in our laboratory, we go straight from the fracturing to the shadowing event, the platinum and the carbon. But if you want to see cytoplasmic and extracellular molecules suspended in 3 dimensions, you evaporate, you sublime away the frozen ice down to some depth, and it will leave, because the molecules leave without ever going through a melting phase, they leave the molecules in 3-dimensional lattices within both the cytoplasm and the extracellular space. So we call it freeze etching if you interpose step E, or freeze fracture if you go straight from D to F. And then finally, for the first 50 years of freeze fracture, the object of the game was to get all of the adhering biological materials off so that you could get the electron beam through the sample and resolve the sample in 3 dimensions. You notice there's a 3-dimensional contour here in the electron microscope. That changed with frill. We'll be back to that. So again, if we go back to how we visualize the replicas, 99% have been examined by TEM. But because SEM prep does not have to digest away the tissues, a few people have tried looking at immunogold labeled samples for scanning electron microscopy. The resolution just was not good enough, so it was not used very much. So the object of the game at first, for the first 30 years of freeze fracture, was to determine where the fracture plane is and to identify the structures that you could see. Now, there were 2 major hypotheses for the first 20 years of freeze fracture, and one was and Mullethaller's hypothesis is that the fracture plane went along either the external surface of the membrane or the internal, the cytoplasmic surface of the membrane at the lipid-water interface. The other idea 10 years later that was demonstrated is that actually membranes are split, and there's a whole history there, but Pedro Pinto da Silva, and by the way, his last name is Pinto da Silva, not da Silva as he's written up sometimes. He's Pedro Pinto da Silva in 1971, and we will show you his images momentarily. He hypothesized and revealed that the membranes are split down the middle. So that early work in the 1970s by Dan Bratton's laboratory, Pedro Pinto da Silva and others, revealed membrane splitting, provided evidence for the particles being protein, and led to the fluid mosaic integral protein model of the membrane, which is still current, the Singer-Nicholson extended model. Pardon me for playing with my screen a bit. Okay, the fractured membranes exhibit abundant intramembrane particles. They're present, the particles are present proportional to the membrane enzymatic or physiological activity. And so it was assumed from the very beginning that these somehow represented the biologically relevant molecules. But there were an awful lot of them and a lot of different kinds. And so it looked to be very difficult to sort out which particles were what. But a few early experiments, literally going back to the early 60s, were to use protease to digest membranes and remove the particles that showed that they were protein, and then to add back to purified lipids to add purified transmembrane proteins, and particles appeared in membranes after the addition of the protein. So it was pretty well established by 1965, 69, that all the structures we were beginning to see in freeze fracture would have biological relevance. We just didn't know how many different molecules there were. So from the very beginning, even before some of the ones that I'm going to show you, there were approaches to freeze fracture and freeze etch labeling. And so here is Pinto da Silva, Douglas, and Bratton's 1971 paper. Pardon me while I have to play with my own screen. This is what they would have called pre-fracture labeling. And in fact, in the sequence, they called it label fracture, label then fracture. On the left, this large, do you have my arrow? Does it show up now, Naomi-san?
Yes.
Okay, excellent. It was gone when you were talking. Okay, so this is a red blood cell freeze fractured. It's very poorly prepared. We didn't really know how to prepare samples back in those days. It was fractured to this thin line you see here. And this would, if you had made a freeze fracture replica, this would be all that you would see. And then you would have seen extracellular eyes. But they then deep etched this sample and they lowered the water table from here to there. So this is an etched outer surface of a red blood cell. And before they froze this sample, they put in ferritin conjugated antibodies to get against the red blood cell A antigen. And what they saw was that on the etched surface, there are these large bumps. And in the fracture plane, there are these other smaller and more distinct bumps. These are the intramembrane particles clustered because of phase separation. And what they were able to show is that all of the ferritin molecules were on the surface of the membrane and not in this area here. And that there was a distinct step from the etched surface to where all the particles were. And that, among other things, allowed them to propose a model much like this, that membranes were split down the middle, that they revealed intramembrane particles and pits, and that the labels were on the external surface of the cell. So this would be the ferritin antibodies attached to the external surface, and that fracturing had removed the outer half of the membrane, leaving a little step at this area from the surface down to the split membrane. And so that's the step is shown here. So one always sees in freeze etch this step. In freeze fracture, you seldom see that step. So let me see. So this was among the first attempts to develop labeling for freeze fracture. And Pedro abandoned this approach for about 15 years until we yelled at him. He had to get back to it. We were doing labeling at the time and getting some success. And I'll show you a few of those in a moment. So early approaches to freeze fracture, also early on, 1987, Griders in New Zealand had done some thin section labeling of gap junctions from cow. This is a cow lens preparation. And what Griders recognized was he could do a sort of like a block preparation. He opened up a cow lens and pressed it onto the surface of an ultra clean glass cover slip. There was a little bit of adhering material there. He then took that to the freeze fracture machine, fractured it, and then put the and shadowed it, and then labeled it with antibody to what was called MP70. We now know that to be Connexin 50, and showed that the immunogold followed the surface, the contour of the gap junction. Here it is fractured and replicated. And over on the right side, as they pointed, they selected an area that had a big piece of debris sticking up in space, casting a shadow where there was no carbon. So the block was over here in the right corner, and the unshadowed area is here. And you see that as this margin where there is the protein barely resolvable here of the gap junction and the immunogold labels all over there. He called this technique frill for fracture replica immunogold labeling. So that was in 1987. People were working on these kinds of approaches. During this period, we were also trying to develop freeze fracture labeling techniques, and this is one of the many different failed approaches that we had. Here's a spleen pellet with white blood cells and red blood cells, and a freeze fracture replica of the surface of that looks just like this. This is one of those preparations. This is immunogold labeled. And what we did was incredibly laborious, but we cut thin sections not perpendicular like this, but parallel to the surface, or as near parallel as we could. And we got thin wisps of replica that we could look at. And we could, in this case, we were able to localize immunogold antibodies against IgM on the surface of certain white blood cells in the spleen preparation. And there were a number of other things that we tried, including shadowing and showing that if you shadowed and did thin section immunogold labeling, or freeze fracture replica immunogold labeling, you could show that the gold beads were always in the unshadowed side. So the replica was some kind of barrier to penetration of the labels. But what we were able to get is very low background, and I'll show you what the problem is in the next slide. So this is why it's important to report failed experiments. This was an absolute disaster experiment that we wanted to share with people. And what we were able to show is that molecular adsorption is a basic property of platinum and carbon replicas to both the platinum and the carbon. This sample is one of those spleen preps that we labeled. And this is the replica running through the sample. And this is a higher magnification image on the right. But notice there was no primary antibody applied. This is only the secondary antibody, and the gold beads are incredibly densely packed without any signal. This is all noise. And there are approximately 10,000 gold beads per square micrometer on the extracellular space where there are no cells, on the inside of the cell membrane, on the outside of the cell membrane. You don't see it here, but we did stereoscopic imaging, and we could show that these gold beads were both on the platinum side of the replica and on the carbon side of the replica. So we recognized that, like platinum is used as a catalyst in catalytic converters for a reason. It's highly adsorptive, thereby concentrating the contaminants, which can then be burned off or digested or otherwise treated. And activated charcoal, which is essentially what pure carbon is used as an adsorbent also because of its adsorptive properties. So we developed an approach to eliminate this source of noise. This is just a replica of water. It was faster than digesting and sectioning tissues. And we had noticed that it was a really good model for showing the same ultra high density of binding. And Tim Johnson, working in my laboratory, was able to develop what we call labeling and blocking buffers, LBBs, which reduced or completely eliminated the noise. This one should look just like that, but in fact, there's no gold beads. And what that was he found that fish gelatin digest, many others have been used, serum albumin, other things, but we found that fish gelatin digest, 10,000 molecular weight cutoff was a perfect blocking buffer. The small, medium, and large diameter peptide fragments would bind to the adsorption sites all over the replica, leaving just enough space between them that antibodies could reach in and grab, and you could have specific labeling. So we published this as we're at the end of our rope. We can't go any further with this, but we've at least solved the problem of non-specific noise. Kazushi Fujimoto read our paper. I talked to him at Cancun a few years ago about how this was done. And he said immediately, as soon as he saw it, he knew what to do. He went back to the laboratory and he introduced the SDS-FRL, which was to use, instead of digesting away all the tissue remnants with caustic things that we had used, like bleach or chrome sulfuric acid or hot fuming nitric acid, pretty caustic reagent, he would use a very gentle way to wash the samples. And so he chose SDS detergent. And that single advance left the technique that we all now use. This landmark paper showed he used rat liver. Let me back up. And this is some of the first images that he published. This is a gap junction in the liver. And so is this little tiny one up here. And I just want to zoom in on this, if I can. Do I have control? Yes, I do. So look at the resolution, look at the specificity of labeling. There's no noise. There's all labeling. Labeling is both on the P face and on the E face. And this was thought to be impossible until Kazushi showed how this could be done. The E face is equally well labeled as the P face, and the specificity is such that even that tiny little gap junction in the upper right is well labeled. He used antibodies to both connection 32 with 2 sizes of gold and connection 26 with another size. And notice there are 6 nanometer, 12 nanometer, 18 nanometer, let's see, 6, 12, and 18. Here's a little larger 18 nanometer gold. So he was able to say that both connection 32 is in abundance, but there's also a fair amount of 26 in the sample. He used antibodies to a variety of cell structures in the liver, including antibodies to occludin. I hope you don't have things over your screen like I do. So occludin, is that bar on your screen? Is there a bar here on your screen?
Nope.
Okay, good. So it's just on mine. So here are tight junctions in the liver, beautifully labeled for occludin 1, showing again specificity with entirely different antibody. And one can do antibodies to totally different things. He could have used 32 in occludin or 26 in occludin. The connection proteins are labeled beneath the particles where you would expect them, and between the and below the E-phase pits, which is a form of so-called cryptic labeling, where the structure that's being labeled may not even be visible as the way you expect it. So we'll go further forward. So Fujimoto explanation is in the original paper is as follows: that the freeze fracture replica, let me see if I can, is made with the dark line here is a 1 or 2 nanometer thick layer of platinum and put on at an angle so that it casts a shadow beyond the particles, and that's the way you resolve the dark side versus the bright side of the particle, and a carbon film thickness over the top, which is 10 to 20 nanometers thick typically. Nanometers, okay. The radius of uncertainty of the labeling is one of the major limitations now. It's not a major limitation in that they're pretty damn close. So in this case, he uses a 20 nanometer gold bead with the center of that gold bead is held away from what it is labeling by the primary antibody and the secondary antibody on the gold, the sandwich technique that all of you are aware of. And this radius of uncertainty is at most 20 to 30 nanometers. But in fact, because these antibodies are in a hemisphere over the around the entire particle, most of them are well within the radius of uncertainty, usually essentially on top of the particle that they label. And the upper surface is the source of non-specific binding, both there and once you have the tissues digested off with SDS, also on the bottom. And notice that there is none. And so I don't remember, I think he used bovine serum albumin to his blocking buffer. Any number of molecules can work. So the technique, and this is also from his paper, Fujimoto's paper. Here's a sample, a slice of replica on a copper hat that he fractured, floated it off. The tissue is still present, and the SDS digestion floats this ultra thin replica off, and the replica is very, very fragile. So he had to pick it up on a large opening grid with a layer of Lexans to support the sample. Notice it's supporting way out in the middle of the holes. Now is a useful technique. Let me back up. That's a useful technique for liver or some homogeneous tissue. But all you guys are interested in the brain, spinal cord, nervous tissue, which is far more complicated than liver. There are far more cell types. And so while Fujimoto was developing SDS-FRL, we were developing something that we called grid map freeze fracture. And that was, these literally were overlapping in time and space. Here is a thin slice, about 100 micrometer thick slice of rat spinal cord in the lumbosacral enlargement. This is a different sample, but we photograph them. Typically, Tom Yasamura, who you'll meet later, will photo map them by confocal microscopy before they're fractured, fracture them, and then explain the preparation here, and then go back to the comparison. So this is a sample. This is a cervical enlargement image of rat spinal cord. It was fractured, and while it was still frozen, a droplet of Lexan plastic held in a droplet on a gold grid was placed on that sample while it was still frozen. The Lexan dichloride solution freezes immediately, and then that's placed into the freezing compartment of a refrigerator and warmed up to about minus 25 degrees centigrade. It does not thaw out, but the Lexan in the ethylene dichloride solvent evaporates at that temperature, and so the grid dries down onto the surface of the replica. The replica is then taken out and thawed and photo mapped by confocal microscopy to the limits of resolution of confocal microscopy. And that's what you see here. That is the gold grid. It's a finder grid, so we can re-identify every single location in that sample. The fracture plane went out of the edge of the tissue over here, but the replica actually went over the entire surface. You cannot see the replica by confocal microscopy except using certain reflectance modes. So this exact area of this exact grid was found in the electron microscope in this area in the lower left. And that is the central canal of the spinal cord. Here's the central canal here. And these are the ependymal cells lining. We were interested in determining what the composition of and the location of these gap junctions. And I'll show you those pictures in a moment. So we then go from this area here. There it is at higher magnification. And still this area is shown at higher magnification in the lower left, showing that we can resolve quite well the gap junction on the surface of the ependymal cell linking to ependymal cells. You also can see the microvilli, and here are cilia out in the distance for markers that you can confirm the identification of cell types. I've got to move this. I can't see my own screen with this. Okay. And now I am locked, apparently. What is the new? It says new share, and Naomi, I've lost control of the screen.
Can you try to unshare and reshare again, maybe?
Okay, it says new share. I don't know what that does. We'll try it here. It says your screen, stop share. Let me just, okay, it's working again. Good enough, thank you. So with the publication of Fujimoto's paper, we decided to combine SDS-FRL with GMFF and decided it was time to go back to Greider's original terminology, which has precedent by 10 years, and call it frill. So here is a sample, one of our labeled samples using the combination of the two techniques. This is the optic chiasm. These are the superoptic nuclei on either side. And the area that's colored in blue is the ependymal lining of the third ventricle. The area that is shown, and then behind there you can see, I've just emphasized that there's always present the gold grid, the gold finder grid, so that we can go back and find the same things over and over again. An image from area CD is in images CD to the right. So at fairly low magnification, we can easily see the 20 nanometer gold beads, find things very, very quickly. These clusters of gold beads are actually on identified gap junctions. And these were labeled against connexin 43. So we were able to show that connexin 43 is present in ependymocyte gap junctions and astrocyte gap junctions, but not in neuronal or oligodendrocyte gap junctions. this also illustrates something else. This image on the lower left is not from the same pardon me for housekeeping duties here. So this image on the left is from one of those spinal cord samples, but it illustrates the point. They are autofluorescent. There is a substantial amount of fluorescence, autofluorescence in any tissue slice, and that is a major problem for high resolution imaging that we will show some images from Jim Nagy to explain this. It's always present. Second point I'll make here is that one cannot use glutaraldehyde fixed tissue for SDS frill. And that is because it's so rigidly cross-linked, even after 1/100th glutaraldehyde for 2 minutes, that you cannot digest the tissue. You cannot wash the tissue away with detergent. So you can use formaldehyde fixation or ultra rapid freezing, but you cannot use glutaraldehyde for any of these techniques, unfortunately. Okay, when I move this off, I lose control of my screen. So let's try again. I'll just try to put up and remember what I've been seeing. Again, Naomi-san, your system won't let me control it again.
We see your screen.
Yes, but I cannot. Oh, okay, now it's working again. Thank you for whatever you did. So the technique that we're doing now, that we now have renamed frill again, is we start with a slice of tissue of uniform thickness. And the uniform thickness is so you can map it before you freeze it and fracture it. And then you can choose a depth within the tissue that you would like to cleave. You can direct the fracture plane plus or minus perhaps 10 micrometers and cleave a specific selected cell, which we've done and published. You cleave and replicate. You then put the gold grid in the Lexan onto the tissue while it's still frozen, air dry it down, and so you draw the final sandwich is going to be gold, Lexan, replica with tissue adherent. We then wash the tissue away with the SDS detergent, but we typically add an additional step. So that's why it says they're either 9 or 10 steps in the process. So we always, Tom Yasamura always, after the replica is made, he will photo map it again, determine where the fracture plane went through the tissue, and be able to map it back to what's been removed from the original slice as step 1. The tissue is washed away with SDS detergent, sandwich labeled with immunogold. You can use a 1-step or 2-step process. We always use the 2-step process. And blocking buffer is critical from this point, from the time you wash the tissue until you dry it for looking in the electron microscope. The labeling blocking buffers must be present. Once it's been labeled, you then rinse and air dry the replica with the immunogold standing up. It's flipped over, you see the gold grid below. And we apply an additional carbon coat to anneal the cracks in the replica, because you've warmed the replica up from -100 degrees in the freeze fracture machine after it's made to +20 or 30 degrees or +70 degrees with the labeling techniques going on. And so cracks, expansion cracks from thermal expansion are a real problem. So we put another layer of carbon over to fill the cracks and also to lock the gold beads in, because reviewers always want to know why they don't move later. Okay, then you remove the Lexan layer and take it to the electron microscope and examine it by TEM. And replicas can either be, if they're terrible, you throw them away in 5 minutes, but many of our replicas were examined up to 50 hours per replica, meaning a whole week or 2 of work, or in Naomi's case, 3 or 4 days of work. She worked late. Okay, so one of the questions is, how much biological material remains on the replica after SDS washing? How can we even label it after it's been washed well? So we took several representative but not yet examined TEM-ready samples and stained them as if they were going to be tissue for thin section EM. Now I say they're not yet examined because if you examine them even for a few seconds with the electron beam, there's enough electron damage and the tissue is no longer soluble, it turns to carbon, to charcoal, and is not labelable, and you can't do any of the other steps. So we took it straight from the final wash, and this is what we found. At the top is a gap junction that's immunogold labeled, but what you can see is with the presence of osmium tetroxide, uranyl acetate, lead citrate, we now see globs of lipid caught in the act of coming off the sample when it was dried, and other thin bits of debris. So we had to stain it with electron-dense materials just to see how much material there was. These lipid blobs are a source of of displaced gold beads, which we see quite often. This particular glob of lipid coming out has a gold bead in three-dimensional space, about a quarter of a micron above the surface. So it would look in three-dimensional space. We can identify that as a kind of noise, but it's signal-related noise. That is, there was dissolving gap junction trapped up in this area and labeled. Second thing we did is tilt the specimen to a very steep angle, and you can see the edge of the gap junction and the increased electron density here. So here's non-gap junction up here and the gap junction itself there. And that material is both proteins that you're labeling, but also the lipids of the specific lipids of the gap junction. So let's look at a couple of examples of the kinds of things that you guys would try. Don't need to follow in our steps. You do your own things. So I'm going to show you some, and this is a very complex title, and I'm going to break it down. It's 3 more lines of text in the title. This is the published title. So we were looking at the association of connexin 36 and connexin 45. Does connexin 45 occur in neuronal gap junctions in the retina? And does it occur in association with connexin 36 that we had shown to be in abundance in the retina? So this is the kind of image that we're looking at. And connexin 36 in all of these images are the 12 nanometer gold beads, and connexin 45 are the 6 nanometer gold beads and the 18 nanometer gold beads. So what we were able to show then is that, and I'll show you the evidence in a moment, that gap junctions that have connexin 45 in them always have connexin 36, well, 90+% of the time, and that 36 and 45 are in both opposing hemiplaques. That is, they're in both cells, in the connexons of both cells. And we did that. Naomi did this with using matched double replica frill, and I'm going to show you that in just a few moments, the steps. We were able to show that here's gap junction F. Do I have an arrow? No, I don't now. At the lower left is gap junction F and G just beyond it, which are shown on the right. And we were able to, with this technique, show that I've got my cursor back. So here's F and G, and this is the matching complement replicas. One is from this set of slices, and these are the split slices from the other side. These are strips of retina. And we have labeling for connexin 36 with the large gold beads and connexin 45 with the small gold beads. So they formed bihomotypic gap junctions. I'll explain that in a moment, that term. And we were also able to show that they had scaffolding contributed by zonula occludens 1, a cytoplasmic protein. And so here's the stereo images of gap junction F and F prime. And notice that what we can see is that connexin 36 is opposite connexin 36 in the complement, and connexin 45 is opposite 45, and we map the areas of the replica that are labeled. We could also tell noise, that's a gold bead, noise gold bead, as is this one up here. Because we do these in stereo, we can see that those 2 gold beads, but none others, were on the wrong side of the replica, where they could not possibly be labeling. So let's see this technique a little bit more, because you're very likely to use this technique a lot. Here is a double replica device. This was done in the Balzer's freeze fracture device in Shigemoto's laboratory. I think Dr. Shigemoto is in the list of participants. So here's the double replica device. with the slots where the gold grid gold hats go in here. Here it is opened. After it would be opened. This is the open sample with samples present that have been shadowed. And this is one grid would be the B sample here is here placed on a gold grid, not a grid in this case, it's a thin bar, they still call them grids, a thin bar grid with single bars, and you can see the bars, and the B prime sample is the other side after the sandwich is opened. And we've searched. So Naomi, after she prepared these, shipped them from Japan to me in Colorado. And we then mapped a couple of hundred of the gap junctions present, literally several hundred, as you'll see in a few minutes. And we first searched one, in this case B, and we would find gap junctions. And here was one, the few percent, that roughly 10% that contained 45. And then we would map that. We would have photo maps beside us at the console of the microscope, mark the location. And then after we'd mapped maybe a dozen of these, literally a dozen, we went to the other grid and then found the same ones in the other complement. We lost a few because there are grid bars that are in the same location in both of them. And there are major areas that we get to look at in both samples. So we do the dotted lines to show where we don't need to look because we'll never find anything. We won't find the complement. So we were able to find the complements in this way. And we were able to distinguish between many different proposed models. Almost all of these had been proposed by one group or another to occur. So the first thing is we found most gap junctions contained connexin 36 without 45. A huge number of them, but a small number of them had 45, and all except one of those had 45 opposite 45 in the other half, as is shown here, and 36 opposite 36 in separate domains. And so we were able to determine that they were not, as we would say, they're not heterotypic. That is, we don't have 45 opposite 36. We didn't find 45 by itself. We didn't find 45 with any other connexin, that 45 was always opposite 45 and 36 opposite 36 in those that had both connexins. And none of these other combinations where we have mixed 45 opposite 36 intermixed, or 45 within different connexins of the same gap junction. So of all the choices that were out there, the A and E were the only ones that we could find. So this eliminated a whole series of proposals that were in the literature at the time and were each equally potentially important. So the next stage is that Frill revealed co-localization of intramembrane and cytoplasmic proteins. So in this particular one, they're labeled for both 45 and Z01. We found 45 is in this case, the 6 nanometer and the 18 nanometer gold beads, and Z01 were the 12 nanometer gold beads. There are only 2 here, but this one below has more labeling for Z01 than it does for connexin 45. Here again, labels for connexin 45. So it can be used for a combination of labeling cytoplasmic and membrane structures. And since most of the gap junctions in the retina did not label for 45, we were able to use Z01 to find gap junctions that presumably are all 36. But Z01 is a scaffolding protein for gap junctions, not associated with a specific kind of connexin, or not with all of them anyway. And it's now known from any number of other approaches that Z01 is a McGuck membrane targeting protein. So I think we're just about on schedule. So here's an example of why rushing to be first can derail your career. So there wasn't, back in the early 90s, there was a rush to be the one to identify the first neuronal connexins. There was a time when we did not know that there was a different connexin than the ones that were in liver. And one of the people who did this, she came into my laboratory for a while, is that our perspective is, and I'm going to show you why with these few images, that in CNS tissues, you cannot be certain of assignment of a particular connexin to a particular cell type by confocal microscopy, possibly by super resolution microscopy. Here's one of Boggy's Nataraja's first images published in 1997 of connexin 32. That's what they call antibody to connexin 32 in supposedly in neurons in a brain slice. I don't remember the particular brain slice. But the problem is will become obvious in a moment, is that in the brain, 90% of the cells are glial cells and red blood cells and are not neurons. And yet you see nothing indicating where the glial cells or the capillaries are in this sample, for example. The second problem is that there's no structural marker to identify a gap junction. And the third is that these areas that are immunostained green and shows up as yellow because of the overlay with the red, is that it all appears to be extranuclear cytoplasmic, which is, no one has ever shown that to occur. There's such little in the cytoplasm being transported that this image itself is questionable without any other data. But the other problems are shown in the next couple of slides. The limit of resolution of light microscopy in the near ultraviolet is shown by the blue dot here. It is drawn proportional to size, and the limit of resolution in the red wavelength is by the red dot. Notice that even the blue dot overlaps with the plasma membrane of this neuron in red, two plasma membranes of this astrocyte process, the outer membrane of the oligodendrocyte, and its inner and outer nuclear membrane. That is the problem with light microscopic localization. To illustrate that a little bit better, the limit of resolution is determined in frill by the radius of uncertainty, as we call it. And this is a freeze fracture, a typical low magnification freeze fracture image where you can barely recognize the E face and the P face of a gap junction. Immunogold labeled for both connexin 26 and connexin 30. And what we see here is the astrocytes, which we identify by structural markers, are in blue, and the two neurons are false colored red. And if that smallest voxel, volume pixel, in the light microscope is illustrated by this block. It's actually twice as tall as it's wide. And the immunogold beads are very close to a resolvable gap junction, but this is the limit of resolution of light microscopy. A photon arising from a fluorescent label on the gap junction could not be localized any closer than the blue dot. And the plasma membrane of the neuron, or the cytoplasm of the neuron, cannot be localized any more accurately than the red. So it is not possible to be certain which, if you don't see the other cells, which cell it is on. And even if you could see some blue between them, all you would know is there are three cell types there, or two cell types, three cells, any of which could have been labeled. It's also been asserted that because you can use fluorescence microscopy to track single fluorescent molecules on ultra-thin cell processes in tissue cultured cells, this is just an old image, that then therefore the detection limit by light microscopy is much better than frill. You clearly saw that none of our gap junctions were labeled 100%. But detectability in a single fluorescent slice is not the same as detectability in a thick section, which is autofluorescent. So we had counted a number of images, and we knew that there were a lot of different sizes of gap junctions in the retina, including large plaque gap junctions, large string gap junctions, medium size, small gap junctions, and even smaller gap junctions that are immunogold labeled. And we kept seeing large numbers, and no one was detecting anywhere near the numbers that we were seeing by fluorescence microscopy. And Carola Meyer, at that time, was working in our laboratory, and we looked at connexin 32 labeling in developing myelin, and we could see not only this, I think that's 16 connexins, I don't remember the exact number, but we could detect gap junctions down to 2 connexin pits here, each labeled by a gold bead, and here's a single gap junction with 2 connexins with 2 gold beads. So as the smaller they get, the more easily they're detected by frill, but we probably miss a few. So Jim Nagy and I worked together for quite some time independently at first, trying to realize what the limits of detectability were. So here's an image from Jim Nagy on the left and a corresponding freeze fracture image. We were seeing huge numbers of gap junctions. These are each numbered separately. And I'll just point out, we have 519, 520, 521, 525, 608, 525, et cetera, 609 here. We were seeing large numbers of gap junctions and mapping them. And we were seeing far more gap junctions. So this is the same area of the retina here. This is the interplexiform layer and sublamina 1, 2, 3, 4, 5. And Jim was seeing far more gap junctions out here in lamina 3, 4, 5, and a few small ones in lamina 2, where we were seeing huge numbers of gap junctions. So we're going to compare those two areas. Remember autofluorescence, because Jim was quantifying the limits of limitations. And so he pushed the limit of detectability, I think primarily by turning up the photomultiplier till just before we began to get noise from the autofluorescence. And look at this, this is the same exact section done in stereo. We're only showing it this is actually in stereo too. The area on the left, when you turn the detection up, the photomultiplier up, sorry, just jump forward. This area here has far more gap junctions, as does this area. But in the area where we were seeing large numbers of large gap junctions, there's essentially no increase in the number of gap junctions detected. So from that, I'm sorry about the machine. So Jim was able to determine that whereas before in these images, he was detecting gap junctions to about 100 connexons. I'm not doing anything. I don't know why it keeps doing that. The limit of detectability in normal viewing mode was about 100 connexons, but he would lose detectability at what we estimated was about the 30 connexon limit. Whereas we know that by frill, we were that, number one, that 90% of the total gap junctions are smaller than 100 connexons, and 50% are smaller than 30 connexons. So fluorescence microscopy was losing at more than half to up to 90%, depending on how you do it. One last thing that I will show you before we close this off. We also use matched double replicas to look at whether, so there are actually 4 connections in goldfish gap junctions. There are 2 homologs, connection 34.7 and 35, and we had suspicions from prior work that 34.7 was not where 35 was. And so this is double replica from that area of that cell, if I remember correctly, that this, what appears to be a gap junction is not, it's actually a nerve terminal. It just keeps advancing. And here the sandwich, the one on the left is the, let's call it F and G are the left and right halves of the same gap junctions, the same nerve terminal. This area is shown at higher magnification here, and this marker, that's a hole in the carbon, in the Formvar support film, just as a marker, and down at the bottom is a crack that shows up in the one on the left. So this is the left complement, and this is the right complement. There, in fact, were 19 gap junctions, of which 11 of them are in this particular image. And the one on the left has all small gold for connection 34.7. And the one on the right has all, so that's 5 and 10 nanometer gold. Again, Naomi, did you make this replica? I think we may have made this in our lab. I'm not sure. This was done with Alberto Pereira, so it was probably done in my lab. So what we were able to show is that 100% of the 34.7 is behind this membrane, meaning it's postsynaptic. And this is the P face of the nerve terminal with all the labeling for 35. So that's in the cytoplasm of the nerve terminal. And so we were able to show that these were heterotypic gap junctions, 100% heterotypic gap junctions. So why is that important? Well, the first thing is that this asymmetry apparently accounts for the fact that these have rectified current. That is, that current goes better in one direction or the other. And number two, they're differentially phosphorylated so they can shift from forward to reverse conducting gap junctions under physiological conditions. So this kind of information can be very important to understanding the physiology of these cells. And we'll skip to, so I'm going to quickly give some kudos to the people who are involved here and give you just a short idea of what they did. There's me, you've seen what I do, but Tom Yasamura, who's attending, made, I think, 99% of all the replicas and labeled them in my laboratory. The only ones that he did not do were the ones that Naomi made in Japan when she had to leave my laboratory. We ran out of funds for her, unfortunately. And thanks to Dr. Shigemoto for taking her on and continuing her training, which led her to get to be at the Max Planck. Kimberly Vanderpool, listed as Davidson in most of the publications. Kimberly Vanderpool did at least half of the electron microscopy for the overall projects that we did. She was better at finding labeled gap junctions and other structures than anybody I've ever seen. Jim Nagy, I want to express all kinds of appreciation for Jim. We met at a meeting in Germany about 1998 when the neuronal connection had just been identified, and Jim volunteered to make the antibodies to Connexin 36 and subsequently everything else that we've looked at. He was able to understand, he understood and was able to make antibodies to the sidedness of the proteins, other designer antibodies for us. And those are, I don't remember the name, the company keeps changing the name, but the company was Invitrogen at one time, and Jim can tell you a little bit more about that. And Jim did all of those, almost all of the beautiful light microscopy of the images, especially the retina and things like that, and was instrumental in sorting out the labeling, the limits on detectability by the various techniques. In addition to that, there have been a number of other people that we've shown their images in this. First is Naomi Kamazawa, who's been was with us starting in 2002 or 2005, something like that. She was at Fort Collins, then Okazaki, and now at Max Planck, and partly in Ryuichi Shigemoto's laboratory. Carola Meyer from Saarland University did the studies of myelin connexins. Sharon Nielsen made our aquaporin 4 antibodies. I didn't show you any of the aquaporin 4 work. Peter Agri and Dan Gorlick at Johns Hopkins. As you know, Peter Agri got the Nobel Prize for his work in aquaporins. Eduardo Rosa Molinar, we didn't show any of his images. Mosquito fish connexin, very much like the goldfish connexins. And then Alberto Pereda, all of the goldfish work was done with Alberto Pereda. He did the physiology, the biochemistry, and we just did the picture taking. And I don't mean to imply that that's not an important part of this research. Okay, thank you very much. I think we've covered everything we need to for the night or day.
With English, I learned from you pretty damn bad.
No, not too damn bad.
Not too damn bad.
Okay.
Okay. Pretty damn bad is so bad.
Yeah.
I'm sorry about that.
So,
Okay, so if you have any question, and then, oh yeah, so would you please stop sharing, John, of your screen?
yes, let's see, how do I do that? Stop, no, not stop video, stop share, I've got it.
Okay. So if Zoom attendees or on-site attendee, if you have any questions.
I see Maria Rubio is part of the group. She did some nice work in our laboratory. Didn't have a chance to show any of that either.
That's true. Any questions to John?
Jim, it might be useful to give just a little bit more information than I did about the quantification of labeling efficiency, et cetera, that we were able to do.
Okay, I have about 3-4 minutes before going forward. So Jim, do you want to comment a little bit, please?
Yeah, I'm here.
Yes, now we can hear you.
Okay. Well, you mentioned the antibodies. It's really quite simple. There's not that much to say. As you know, most proteins occur in multimolecular complexes. So if an antibody doesn't work by immunofluorescence, it doesn't mean that it won't work by frill, because sometimes an antibody won't work because the lysines are cross-linked and whatnot, if you choose a poor sequence to make an antibody against. And other times it's simply blocked by dozens and dozens of proteins within a complex, and yours of interest might be in the center. So it might not work by immunofluorescence, but in frill, as John explained, that gentle SDS washing might be sufficient to wash away some of the surrounding blocking proteins and expose your epitope of interest. So,
Of course.
sorry, that's about the only comment I have about it. So if it doesn't work by immunofluorescence, don't give up on frill necessarily. Otherwise, everything else, fluorescence, immunochemistry, or whether antibodies used by frill or fluorescence, everything else with respect to antibodies is pretty much the same. And whether you choose an antibody against an extracellular epitope or an intercellular epitope, labeling the E face or the P face, it really doesn't make much difference.
Hey, thank you, Jim, for your comment.
Okay.
Okay, so it's about time to move on. I didn't set up any special break with this small session in the morning, but yeah, please feel free if you need, get out and then come back. So let's move on. So we do screen share back with us. So now we are moving on to more practical part of this workshop. Wait, screen share. Okay.
There we go.
Okay.
Well,
So
they're getting
now,
said. I want to thank all of our participants, the ones from my laboratory. I haven't gotten to talk to you separately with this, but thank you all very much again.
okay, thank you, John. Then now you could mute, please. Okay, so let's move on. So from now, Debbie and Conan are giving a talk about practical part, how to make replica, how to label replica, and then how to analyze images. Okay, Debbie, go ahead.
So my name is Debbie, as you see, and today with my talk, I will tell you a more practical way on how to make and label the replicas. And I will show you what happens when the slides arrive, from the slides arriving to the EM facility to the image acquisition of the antibody label replicas. But then what happens in between? It is sometimes those steps in between that have the little tips and tricks that make the experiment possible. So this is what I'm going to tell you today. When we first get the slices, the brain slices in our lab, we start doing cryoprotection, and we immerse the slice in glycerol. And this is important because we need to avoid the formation of ice crystals that can damage the ultrastructure. And glycerol is a good anti-freezing agent because it forms bonds and that make a less damage to the ultrastructure and don't affect as much the osmotic pressure. Also, another important step that we do for our slices, while they are being cryoprotected, we trim the area of interest. And I usually try to keep some landmarks in these trimming, because these landmarks will become very important later on for imaging. This example here, this is my area of interest is amygdala. So I try to keep the external capsule, which is just a bundle of axons. And as you can see here, this is my trim out, my cutout. I'm only interested in the upper portion of the amygdala, the lateral amygdala. So I maintain this and keep it for further imaging. Sometimes the cells of interest are expressing a type of fluorescent protein, indicating that these cells have some type of perturbation, like in this case, which is a section of the brainstem. And as you can see here, so we have, this is my area of interest. So I trim out only these region, and still, because obviously later on we need to know which one is the side that's expressing. So we freeze them separate, of course, the expression side and the non-expressing side. Okay, so in a nutshell, what is it that how do we prepare the samples for freeze fracture? John mentioned already some of these steps. So this is a reminder, because yeah, this is a different technique that a lot of you might not be familiar. So that's a reminder so that you can remember that technique better. So we start with sample freezing. Once the slices have been cryoprotected and trimmed down, we start with sample freezing. No, we do not freeze on an ice cube tray, even though the diagram looks like a little ice cube. We freeze under liquid nitrogen with high pressure. And then this frozen sample is going to be fractured at the weakest point of the lipid bilayer. And once fractured, the intramembrane proteins will be exposed, and it will give us two leaflets. Like John mentioned, one's going to be the protoplasmic layer, and the other one's going to be the extracellular layer or exoplasmic layer. Once fractured and replicated with the deposition of carbon and platinum, we proceed to do SDS treatment and immunolabeling. So I will talk about each of these steps more in details during my talk. So there's different type of equipment that can be used for freezing samples. There's different machines. This one is from Balsers. This is the HPM 010. This is the high pressure freezing HPM 100. There's this one that's called EMPAC 2. And sometimes there's also the cryoplunge freezing. The cryoplunge freezing might not be ideal for freezing tissues, but if this is all you have in your lab, then it could be used, but it is not the ideal case. Okay, so how do we freeze our samples here at MPFI? So this is the machine that we have, is HPM 100, and we use 4.6 millimeter carriers that are made of copper coated with gold. And we add a spacer of 150 micrometers thickness of double-sided sticky tape. And you can see here in this space, this is where our tissue is going to go. And our tissue are vibratom sections. And these sections are between 130 and 150 micron thickness. And as you can see, that thickness matches the thickness of the tape. This is important because we obviously don't want to squeeze the tissue, but we want the tissue to have good metal contact with both sides of the carrier. So you can see here, this is the double-sided tape, and in the space in the middle are my pieces of tissue. And this is going to be covered by the piece of another carrier that does not have the tape. And then it will be inserted into the ceramic plate. It'll be ready for freezing. Ideally, it would be better if I have the entire area with tissue, but it's not always optimal because of the shapes, or I don't want to make them super tight that then they will be all squeezed in there. So I use some glycerol to fill up the space. Again, and then once it's frozen, it comes to the star of our show. our freeze fraction machine. And we have a JFD2 from JOL. And this machine has 2 separate chambers. It has the sample chamber and the evaporation chamber. And this is where the carbon and platinum rod are located. And here we are in, when I'm still learning freeze fracture from Naomi. So these are the tables, that's how we call them, where the carriers are going to be inserted. These are the 2 that we have in our lab. We have a double replica table. This slots is where the carriers go, and this is a single replica table. Mostly this one we use for cells that are grown on either a grid or sapphire disc. Mostly we do tissue, so this is what we use on a daily basis. Either one of these tables can be inserted in this rod, and this is how we insert the sample into the freeze fracture machine. So, like I mentioned, this slots is where the carrier go, and as you can see in this video, the sample is already in there, and I insert the table into the liquid nitrogen to cool it down, and it's only until it stops boiling that I will insert the carriers into the slots. Now, since the sample is already frozen, so it's important that we do everything, keep everything under the liquid nitrogen temperature, because we don't want the sample to be affected. And so once the table is cooled down, you can see in the video on the right side, I proceed to insert the carrier onto the table. Notice that I am leaning the table against the wall of this box. And that's important because once the sample is inserted, we want to be extra careful not to open the table, because if we do that, then we will fracture outside the machine and it will defeat the whole purpose. So yes, it needs a little bit of practice to be able to do it more smooth. So what's next? So after we insert the sample in the carrier, we proceed to insert the rod into the freeze fraction machine, as you can see here. And now this is under vacuum. So now we cannot just insert the sample, because obviously we come from atmospheric temperature pressure. So we have to leak the vacuum a little bit. We flood it with nitrogen, and until the chamber is ready, I open the latch to insert the sample. You can see here the table is being inserted, and it goes inside this shroud. And this shroud is a contribution actually by Dr. Rush. And this prevents water contamination once the sample is fractured. And so now that the sample is inserted, we adjust a knife. This is a knife that will fracture the sample. And you see that this pin here, the knife will hit that and make the fracture. You can see the knife moving forward, and the table is open. All these under vacuum. And now that it's open, the intramembrane proteins are exposed, and then we start rotating the table, and I start adding current. And you will see up here that the filament is shining, is red, and then you will see the rod. In this case, is the carbon rod. And you will see that the table will start glowing. That means the carbon is being deposited onto the newly exposed intramembrane proteins. We start, and this is how we start making the replica. We add a very thin layer of carbon first. This is to stabilize the newly exposed proteins. And after we add carbon and platinum, this is how the replicas look like. As soon as we take it out, we take the table out of the freeze fraction machine, we put them on a ceramic plate containing PBS. At this time point, they can come to room temperature, and this is what they look like. Remember, I said I always keep landmarks to do better orientation when imaging. So you can see the color is a little bit grayish. So this color is given by the final deposition in the sample was carbon 3 nanometers, platinum 2.5 nanometers, and 35 nanometers of final carbon to stabilize the whole membrane. So once the replica is made, we remove the replicas out of the carrier and we put them in these vials containing SDS. And then we go to do digestion, and we digest in oven with at 80 degrees, at 82.5 degrees in our case. And this is because we calibrated our oven and we concluded that it's equivalent to 80 degrees. And we do agitation, and we digest for 19 hours. And you can see here, we have a little camera because during COVID, we needed to keep an eye, and we were not always inside the lab. So we needed to, as everybody else, do some adjustments to our protocols. And so once the replicas are digested, we again take them out of the glass vial and put them in the ceramic plate. We use a ceramic plate because the replica will not get stuck. And we use a platinum wire loop to move them around. And now I add washing buffer, which contains TBS and a little amount of protein. In this case, like Dr. Rush mentioned, we use fish skin gelatin to already start the process of blocking, and at the same time to have protein as soon as the samples come out of the SDS solution. And like he mentioned before, the SDS solution basically works like in the Western blot. It's acting on the proteins to unmask the epitopes that we're going to use. So once they come out of the oven, now we have the epitopes exposed, and we don't have all these other tissue that we had. And now we can proceed and do antibody labeling. And this is how I do my labeling on a plate that acts as a humidity chamber. We do the labeling at room temperature on the shaker. And just like in confocal that you can use 2 or 3 different primary antibodies, we can do the same. In our case, we can do them all at the same time, meaning we mix them all in the same tube, or we can do them consecutive. And once we do the primary overnight, we add the secondary. You can see the drops look pink, and this is because we use colloidal gold, and they have the colloidal gold is reddish. And the same thing, if we use 2 or 3 primary antibodies, we can add them all together, and we have all the secondary antibodies in the same drop at the same time. Keeping the replicas intact, like Dr. Rush was mentioning that in his lab, they use a technique using Lexan. We're not using Lexan, and so sometimes keeping the replica intact can be challenging. In this case here, you can see the replica is intact coming out of the freeze fraction machine, but sometimes after digestion, this is not always the case. And so you can see here, this one, it's pretty good after once digested and handling. That's when sometimes the replica break, and this one is pretty intact. So that's good for later on for imaging, especially when we're trying to image things that we need to have layer information, like in the case of the cerebellum, where we're trying to follow the dendrites from the Purkinje cell. So having the entire molecular layer is important so that we can follow dendrites along the molecular layer and be able to determine, for example, distribution of a particular ion channel like calcium. But sometimes, like I said, keeping the replicas intact is not always possible as much as you try, and they tend to break down. So this is when having landmarks, inherited landmarks on the tissue is important, like the axon bundles that I was trying to keep in when I trimmed out the slices. In this case here, remember we kept the axon bundles for the section of the brainstem. In this case, we're examining of the MNTV. And yes, the cells have fluorescence, but obviously we don't have fluorescence in our EM samples. So you can see here, again, we have the axon bundles, and we can see these large circles are the somas that are our cells of interest, one of the cells of interest. We're interested in the synapses that are made to the soma. And this is what they look like. We are interested in the synapses that are made by the calyx of health. You can see is this, hence the name is this calyceal shape cell engulfing or involve the primary neuron. And you can see here, this is the principal neuron, this big circle here. And this color in blue is the calyx of health terminal. So it's equivalent of this orangish yellowish. And we know that this is the site of the synapse because we can see that is contacting glutamate receptors on the postsynaptic E phase. And on the presynaptic side, we have cross-fracture of synaptic vesicles. And in this case, we have two different sizes of gold particles, GFP to identify the cell of interest. This is the cell that we are interested in imaging and that has some molecular perturbation. And we're interested in seeing the calcium channels. In one of the first examples that I show you that I was using lateral amygdala. This is my region of interest. And in this case, I don't have a specific marker to identify the cells of interest, but I'm just going by the region. So hence important to keep the landmarks. I'm interested in the cells located at the very apical portion of this triangle. And because I have these landmarks, I can see them again in my replica, and I can go directly to the region of interest and know that that's the location of my cells. And this is what the cells that I am labeling in amygdala look like. So in the first example of labeling, I show you, I had two different sizes of gold. I had GFP and calcium channels. So in this case, I have three different sizes of gold particles. So just like in the confocal that you can do multiple labeling, same thing, we can use multiple antibody labeling at the same time. So I have 18 nanometers to identify VGLUT2 and the presynaptic side. And so this is P phase of the presynaptic side. And this is the postsynaptic side. And this is E phase. And on this side, I have 12 nanometers to identify AMPA and 6 nanometers to identify NR1. So we know that this is the side of the synapse because we have direct contact with the presynaptic side. And in this case here, I actually was a bit more adventurous and did 4 different antibodies at the same time. So yes, these humongous gold particle might not be very appealing. So this is 40 nanometers for VGLUT2. And I have, in this case, I do have GFP with 12 nanometers. And these other small shape is VGLUT1. And again, I have PANAMPA on the E phase of this postsynaptic region. So now we have all the images that we need for analysis. So, okay, so now what's next? So that's the fun part. The analysis is fun, interesting, and that is what Conan is going to talk to you about next.
Thank you, Debbie. So we're gonna, I think, hold questions until after my talk. So just go ahead and do this. So hello, everyone. My name is Conan Thomas. I'm a research assistant in the EM core facility. And I'm going to talk to you today about how to analyze these labeled replicas. So Debbie just nicely showed us that we can label and create these replicas. We can label them with different sizes of gold particle. As she mentioned, we can identify the pre and postsynaptic cells based on labeling. So here we have a glutamatergic synapse where we have VGLUT2 labeling this axon, and we have AMPA and NMDA receptors labeled with 2 different sizes of gold, which show us the area of the synapse. So next, the question is, how and why can we use freeze fracture frill to do some sort of quantitative analysis? There are a few reasons why frill lends well to quantitative analysis. One of the reasons is that we get this really nice, unique 2D view of the membrane proteins and their arrangement on the membranes, and we can analyze that. Frill also has a higher sensitivity than pre and post embedding labeling, which are other techniques that we can use in electron microscopy to analyze and label proteins. There are some considerations we have to take into account. For example, labeling is not necessarily 1 particle per 1 protein. There could be 2 particles or no particles marking a protein, but it does serve as a proxy for basically quantifying the amount of protein. We also have to think about labeling efficiency. So this is variable by antibody lot, concentration. If we use different sizes of gold particles, it can change, and also SDS digestion condition, etc. So there are a lot of things that we have to keep in mind when we do this type of analysis. So let's think about what kind of measurements and basic questions that we can ask. Let's think of an example. Let's imagine we have 2 animals. One of them is a control, and the other one is a mutant. And in that mutant animal, we have synaptic proteins removed from some synapses. What are some basic questions we can ask? We can ask, do synapse sizes change in a specific brain region? Does the density of AMPA and NMDA receptors change if we're looking at glutamate receptors, glutamatergic connections? What is the ratio of AMPA receptors and NMDA receptors? These are all sort of basic questions we can ask. And none of these are necessarily dependent on the positions of the particles. I could use a clicker counter and count the number of AMPA and NMDA receptors at this IMP demarcated synapse. And I could kind of tabulate this pretty easily. I could pretty easily measure the density based on the area of the IMP clusters. Then the more difficult part is, what if we want to analyze the positional relationships between particles? That requires a bit more information, and it's a bit more complex. So on the left side here, we have a calyx of held terminal. This is a presynaptic axon terminal, as Debbie mentioned. And we have CAV 2.1 calcium channels labeled on this membrane. There are many questions we can ask. One of them might be, how do presynaptic calcium channels distribute at the membrane of the calyx of held? You can already kind of see that they are forming these little clusters. How do we quantify this? So we can ask, do calcium channels cluster together? We can analyze this in several different ways. One of the ways is a measurement called the nearest neighbor distance, or NND. So it's basically looking at the distance between 2 close particles. We could also perform a clustering analysis. There's many different types, but basically we can push them into different groups based on their proximity. We can ask if calcium channels clusters themselves are aggregating. So what we could do is measure the separation distance between the center of mass of clusters. If we sort of zoomed out and looked at the entire cellular level or subcellular level, here's again this example of the calyx of held, we could ask, perhaps there's a subcellular distribution gradient of calcium channels from the axon to maybe the tips of the calyx. And we could ask, perhaps are other active zone components aggregated nearby calcium channels? So we know that calcium is really important for synaptic function, calcium influx, and so perhaps there are other active zone proteins that preferentially locate towards calcium channels. So these are really interesting questions, but to answer them, we need a couple things. We need spatial coordinates of these particles, and we need some sort of algorithms to quantify their positional relationships. So here I have the same region, and this is just a little video. We first have to do annotation of the gold particles. So this is me manually going through an image J and clicking the positions of these particles. This is actually sped up 2 times, so it's a little bit fast. And manual annotation is commonly performed. We've done this a lot in our analysis in the past. And this is something that's reasonable to do if we have like a synapse with a few dozen particles. It's not reasonable if we have really large areas of membrane or if we have numerous images that we want to analyze. So ideally, we can find some way to automate this process, but still maintain a high accuracy that we have by annotating by hand. So thankfully, in the last few years, I'm sure many of you have heard of this new advancement called deep learning. With deep learning, we're basically training a network, or we're training the computer to do annotation and analysis for us. So let me just briefly explain what happens here with an example. Let's say we have some image data and there's some stuff in it. Let's say we're interested in the triangles, extracting the triangles from these images. What we can do is we can create some training data. So we can go through a subset of our images, color in the triangles, we label the triangles in all the images, and then we feed those images into this neural network. And the neural network is just a set of different layers of hidden layers. Some of them are hidden layers, and they're basically, they each have what's called a weight. So as we feed in this training data, the network learns, quote unquote, the features of our labeled objects, our triangles, and stores that information as weights within the network. Then once the network has done this, it's learned what a triangle looks like, when we feed in new images that it hasn't seen before, it's able to produce a segmented output for us. So this is really great for, if we have a lot of images that we don't want to analyze by hand, we can train this network to do it for us. So now I just want to kind of go into some of the literature and how annotation, how automation is being used for particle annotation. So in 2018, Rafael Lujan and colleagues published this paper highlighting a software called GPDQ. So with this software, they developed a method using supervised classification models. So this is machine learning for particle detection, and specifically for one size of particles, 10 nanometer size particles. And they were also able to develop some methods for clustering the particles. And in 2020, work from Riichi Shimoto's group, including David Kleindienst, they basically took this GPDQ software and added some improvements to it, expanded the capabilities. They made it possible to analyze multiple unlimited sizes of gold particles. And they also added this feature that included deep learning active zone area segmentation. This also worked on postsynaptic density areas. And they showed that this deep learning automated segmentation of these active zones was very high accuracy and had a very high similarity to manual annotation. In 2021, around the same time, we were also thinking about this annotation problem. We had a very talented high school intern in our lab who decided that they wanted to develop a deep learning approach to gold particle annotation. So he, Diego Jerez, developed a deep learning network, which is a generative adversarial network, or GAN, which we trained to identify particles. And we were able to discriminate particles by size, specifically 2 different sizes of particle. We found that this had high accuracy and it was able to run on really large, kind of irregularly shaped images, which we work on a lot. It's like the calyx of Held. And it requires significantly less time compared to manual labeling. Since then, recently, we have found some new software called Dragonfly. This is free to use software published by a company called ORS. And this software is amazing. It's a platform for deep learning based segmentation, basically. So we use this a lot for membrane demarcation and particle detection. On the left side here, you can see in blue, these are patches of calyx of Held P-phase membrane that we're interested in analyzing. This is done before we even manually proofread this, and it has a very high accuracy already. We use a linked net architecture for that. And we use a different network, a UNet architecture, for extracting gold particles. So in this case, we have 2 different sizes of gold particle, these blue and red particles here, that we can extract automatically. Now I want to move a little bit into the analysis side of things. So before we were just talking about how to annotate the particles. And I mentioned that clustering is a really important measure for seeing how particles are aggregating on the membrane. And there are many different ways to measure clustering, but I'm going to describe a couple of the most common methods. So the first is something called agglomerative hierarchical clustering, or AHC. And with this method, what we do is we have a set of particles. We draw some circles around them with a set radius. And we start by combining and assigning particles to a cluster if they're within that certain radius. And we start with the particles that are closest to each other. And we slowly group them together based on their proximity. And so just as a basic example, what we get here is these discrete groups of particles based on their proximity. There's another type of method that's commonly used called dbScan. It's a clustering algorithm as well. And it's, I would say, a little bit more conservative in how it generates clusters. So this algorithm takes two parameters. One is called epsilon, which is the set radius that's extended from each of these points, and also this min points. And this is the number of nearby points within that radius necessary to assign a point to be a core point. What does that mean? So you can see in this diagram, we have these red points, and these are the core points. And so basically what that means is if we have a min point set to 3, all of these red points have at least 3 particles next to them within that radius. And we also have these yellow points, which are next to a core point, but they're not core points themselves. So what we can do is we can assign a cluster as being core points and all the ones next to the core points. And then we also have these noise particles that are far away from everything. So here we have just an example of if we tweak the parameters of this algorithm, we can change how our clusters are generated. It can be make it more liberal or conservative in the way that we cluster, that we form clusters, depending on the settings of these. Now I just want to talk about some examples of how clustering analysis is used in the literature recently. So Zoltan Nusser's group in 2017 published a program called Gold EXT. And with this, they were able to basically put together several clustering algorithms. And they used this to analyze RIM1-2 and orexin-1 alpha clustering in the cerebellum. David Kleindienst and the colleagues used DARIA to examine CAV 2.1 clustering and CA3, CA1 synapses in 2020. And again, a paper from Zoltan's group looked at used dbScan to look at clustering of MUNC13-1 at synapses of CA1 fast-spiking interneurons. So this is a very common technique that's used in the literature right now for assessing how particles are clustering. And all of these examples I've mentioned are sort of looking at single synapses. And this is a view of a single synapse. But we often in our lab work with really large fields of view, really large images. Calyx of Held. We also look at somas and lengths of dendrite, which can be really huge tiled images. And so just to give you a sense of scale, if we shrink this down, this is the size of images that we regularly work with. We have all these different patches of membrane that we want to analyze, and there's really irregular shaped images. And this is difficult because the programs that have been developed for synaptic analysis are not really designed for these images. They're really large. And so we had another talented high school student, intern, come to our lab a couple of years ago, Seth Golden, and he wanted to tackle this issue for us. So we worked with him to develop an analysis toolkit that allows us to perform subcellular analysis of these large tiled images. And we made it easy to install and run, no programming knowledge necessary. And also we wanted to design it to have a modular design so we can easily add new analysis tools as we develop them. And so we titled this Gold In and Out, or GEO. And this is a toolkit that integrates different analysis methods into a user-friendly package. And we published this recently in Frontiers in Neuroanatomy, and you can visit our GitHub and download a copy of it. So I just want to go through the interface here. So this is what it looks like when you download an open program. What you can do is upload an image or a folder of images with scale information. You upload a mask that demarcates the region of interest of the membrane. We often use Dragonfly to do this for us, basically. We also have to upload the XY coordinates of gold particles. Again, we use Dragonfly to use deep learning to generate this. And then we can also upload XY coordinates of a landmark population of particles, which I'll explain in just a moment. Then we select the workflows that we wish to run. We don't always want to run everything all the time. Sometimes we might just want to do nearest neighbor distance measurements. So we can select that, only that, and run it. Pressing start. So within Geo right now, we have several different modules. I'll explain right now. The first three that we have is the ones I talked about at the beginning of my presentation: the nearest neighbor distance, clustering, and separation. And I should mention that for each of these modules, we built in the ability to compare a random distribution of particles compared to our real distribution of particles. So we decided to apply this for the calyx of Held, and specifically calcium channel labeling at the calyx. So here you can see on the left, we have an input image with calcium channels labeled with large gold particles. And on the right, we see these blue particles here. These are the calcium channels, and we drew nearest neighbor distances between them. We also have the random distribution of particles with these lines drawn as well. And now we can compare the distances, and we can see very clearly that CAV is clustered on the membrane. We can also perform a clustering method using agglomerative hierarchical clustering and see that we generate clusters with real CAV particles compared to random. Much less clustering happens in the random example. And we can also look at the separation of these clusters. So now I want to talk about a couple sort of new analysis techniques that we included in this program. And these are specific to comparing a population of particles to what we call a landmark feature. A landmark feature could be another population of particles of a different size of gold, or it could be some sort of cellular feature that we want to compare to, like the position of dendritic spines along the dendrites. So this first method we developed was called Gold Rippler. And this provides a metric called the Landmark Correlated Particle Index, or LCPI. And this is a measure of positional bias of particles relative to this landmark feature. I'll explain that. We also developed an algorithm called Gold Star, which measures the nearest neighbor distance between all of these particles and the landmark feature. So just starting with Gold Rippler, I want to explain this idea of this LCPI metric that we developed. So on the left side here, we have a patch of membrane, a circular patch of membrane, and a bunch of particles that are randomly thrown onto this membrane. And they have the same likelihood of being anywhere on the membrane. And in the middle, we have this landmark. If we draw a circle of a set radius around this landmark, what we can do is we can measure the fraction of particles contained within that circle or ripple. And we can measure the fraction of the membrane area captured by the ripple. And if we divide the two, we get the LCPI. So we can calculate this for different radii of circles. And in this first example, we're sort of increasing the membrane and the number of particles that we capture at the same rate. So this LCPI value should stay close to 1. Now, if we think about an example where we have a set of particles that are sort of biased towards a landmark particle, we can imagine that if we draw this circle, this first circle has a lot of particles, but it doesn't have as much membrane area. So that LCPI value is going to be very high. And it's going to stay high, and then it's going to slowly drop back to 1 as we fill the entire area and collect all of the particles. So in this example, it starts high and then approaches 1. And this method was developed by Diego Hedges in our lab. So we wanted to run some simulations and see what kind of curves we could expect to see for different particle distributions around the landmark feature. This first example here, we just have like a random splattering of particles according to a uniform probability distribution. And we looked at both light and heavy distribution of particles. And we can see that this LCPI value, like I said, stays very close to 1. And it's regardless of how many particles we put down. And so this kind of tells us that this method should be resistant to differences in labeling efficiency and labeling density. So even if we have more particles, we should still get sort of the same result. This is what we expect for randomly simulated particles. Now we can think of some theoretical distributions from many different cases. The one that's really interesting to us is this sort of Gaussian distribution, where we have particles that are close to this landmark. If we look at the curve here, we can see that this first circle has an LCPI value of 10 or above 10. That tells us that any given particle is about 10 times more likely to show up in this first bin compared to random. So next we wanted to actually test this on some actual data. And we wanted to basically use an example that we already know two particles or two population of particles are grouping towards each other. And so we used an example that is known to colocalize. So we have calcium channels, CAV 2.1, and vesicle machinery protein MUNC13-1, which are known to colocalize. And we did this at the calyx of Held. And so we actually had two different experimental conditions for this. We had 9-hour SDS digestion and 18-hour SDS digestion. We did this because we wanted to see how differences in labeling density will affect this value, this calculation. And so we ran Gold Rippler using MUNC13 as a landmark. And what we get when we generate this LCPI curve is basically, so on our x-axis, we have the distance from the landmark. And in blue, we have the real distribution of particles, and orange is random. You see that random stays close to 1 as we expected. And the real particles follow the same distribution as our Gaussian distribution. So we found that CAV 2.1 is about 4 times more likely to associate with MUNC13-1 compared to random within the first 45 nanometers. And we saw that this was true regardless of the SDS digestion condition, which means that it seems to be resistant to labeling density changes. Next, we looked at Gold Star. We tested Gold Star on the same replicas. And we found that CAV 2.1 is closer to MUNC13 compared to random, but only following the 18-hour digestion condition. So what this told us was that this method is a little bit more susceptible to variations in labeling density compared to Gold Rippler and this LCPI value. But at the same time, one thing that this technique gives us that Gold Rippler doesn't is we're able to get very concrete distance measurements between particles. So just to summarize and conclude here, I talked about quantitative analysis and how it can take many different routes depending on what questions we have. Deep learning has made our analysis much faster than what it once was. And our lab developed this Geo toolkit for convenient analysis of large images, and we included 2 new methods for analysis. And we're still working on including new features. So we recently added a multi-folder upload system so we can put in a bunch of different images at the same time. And we're working on a new pathfinding algorithm to supplement our Gold Star algorithm. And I'd like to acknowledge the entire lab and say thank you to everyone we've worked with. I want to thank in particular, our collaborators for freeze fracture, Dr. Sam Young, Ryohei Yasuda, McLean Bolton, and Jason Christie for their continued collaboration and fruitful work. And they allow us to continue doing freeze fracture and practice this really beautiful artistic technique. So thank you. Are there any questions?
So now, oh, stop sharing. Okay. So now we have Q&A session, about 10-15 minutes. if you have any question to both Debbie or Conan, this is the time. And then I see some question came to the chat. So first question, I think this one. Yeah. So first question to Debbie is why do you not use Electron to prevent replica fragmentation?
We tested using Electron once. I tested using Electron once, and I wasn't very successful at using it, and I'm really okay at getting intact replicas. So that's mainly the main reason why I didn't use it.
Yeah, I had a similar situation from my Japanese colleague. And then probably we guess in Colorado, we have very low humidity. And then, but Japan or Florida has more high humid. So even we are working in a building, maybe this humidity makes some difference to the attachment of Electron to the replica, or just we have to learn more. But Shigemoto's lab also developed not the Electron, but like a super glue itself to do similar thing to attach the gold index grid. So if it's really, really necessary to stabilize intact replica, we have some way. And the next question is, John commented, I'd like to address accidental prefracturing and thermal stress prefracturing of cell on glass slide. So John, please comment. We are going to unmute. Yes,
Okay, so one of the things that we noticed is that when you first get started, everything works great and the images look fine. And then after you get a little bit of skill, all of a sudden they're all terrible. You can't recognize IMPs, there's garbage everywhere, etc. And then it takes a while to figure that out. And I had another laboratory contact me about this for specific reasons. Why could they no longer see, they weren't even sure they were looking at the proper cells. And what we traced it to was that when you're first starting, you're very, very careful. As you get to be a little bit better, you get a little sloppier. And one of the things that happens is pre-fracturing that you may not even notice. That is, you may squeeze the samples a little too hard or put a little stress on them and they don't fall apart, so you think they're okay. And what pre-fracturing does is that it coats both opposed surfaces with a heavy, heavy layer of water vapor, and that they don't fall apart. You fracture and replicate in the machine, and then they look terrible. And by terrible, I mean at low magnification, it looks like there are cells everywhere. This is mainly on glass slides, but it also can occur with metal, sandwiches. So, but you need to have the sample. It's a valuable sample. So it turns out that once you get a little bit better, then you have to do deep etching to clean up the contaminated slices or surfaces. And so that by letting them sit in the machine for 5 or 10 or 15 minutes at high vacuum, but raise the temperature to -100 or so, they will clean up and they will be good samples again. And then when you shadow them, they will be absolutely gorgeous, except they may have some big chunks of ice on them that won't etch away in that amount of time. So pre-fracturing is always a problem, even if you do everything right, because you cannot cool a sample in freezing uniformly from edge to edge, top to bottom. And so there will be thermal stresses no matter how good you are. So we found that in samples that stick together, that don't fall apart, we would have 10%, 30%, 50% of the area would be beautiful, but 90%, 70, 50% of it would be terrible. And those were always the pre-fracture areas. We published some images many years ago of where how far the fracture plane would go into the tissue. And we called it, I can't remember, something, the high tide mark. It would look, there would be, the particles would be larger, smoother, not as distinct up to the high tide mark, and then they would be gorgeous. So we knew that occurred. And so it was important in the laboratory. One of the people that's here, I won't mention unless they want to talk about it. They were able to get their images by going back and just cleaning this, doing deep etching of freshly cleaved surfaces and get the necessary data. So just want to remind people that no matter how good you are, pre-fracturing can occur. Thermal stresses, which you can't control, high pressure freezing or otherwise. And if you're not real careful and you think, well, maybe it didn't pre-fracture, think that it really did.
I see. Thank you, John. And the next question, can you read here?
What if the replica process is processed with crow-merge compared to SDS cleaning?
Yes.
I don't
I think
know.
that's bleach, one of the bleach and the harsh washing that John mentioned.
Crow-merge is chrome sulfuric acid. No biology remains for labeling. So you cannot use it. It has to be something as gentle as SDS.
Yes.
What surprises me most is how long and how high a temperature you can get away with and still get good labeling. We were so careful at first. Well, should we wash them for 45 minutes or an hour? Should we do it at 37 degrees centigrade or should we go all the way up to 45? And now you guys are doing what, 80 degrees centigrade in 2 days and, go on a vacation in the middle.
Yes. Do we have any other questions?
I had a question for Conan.
Yep.
Yes.
For the analysis of the frill.
Yep.
I was wondering if you were looking at other like positional relationships besides the co-localization, because it is important to know how two are related together, how close they are. But is there other positional relationships that are important to look at?
So mainly we look at, like I mentioned, the nearest neighbor distance. We look at clustering and the separation of clusters. That's mostly, yeah. Did you have another idea for something?
I guess I was just interested, when you said the separation of clusters,
Yeah.
what does that say about the ionization?
Yeah, so it's possible that, because if we think about the function of these clusters of particles, they, if we think about calcium channels, there's a sort of a local influx of calcium into the cell. So if we have groups that are separated pretty far, we might imagine that calcium would be more diffused in the cell compared to if they're all clustered together, grouped together. You might expect that the concentration of calcium to be higher in that region. So it sort of tells us a bit about the function of the cells.
Okay.
Any other questions? Yep, looks like we are good. So I think, at the end of this talk, we have some requests. Let us share our presentation again. Yes. So just for your information, Max Planck Florida is celebrating our 10th anniversary here in Jupiter. So if we miss your Zoom question, we will reach you via email by answering questions. And then all attendees will receive our survey email. So please help us to improve our workshop for the next time. And then actually, we are going to have on-site demo for practical session this afternoon, but our space is very limited. So I'm sorry to say, but I can only have a very small number of people today. So, but we received lots of requests who are interested in on-site demo. So we are going to plan another on-site demo in the near future. And you have any other question, please email me and we are happy to answer. So with this, again, thank you to all of our attendees from all over the world, from very new people for this technology to expert of this technology. I really appreciate. And then we want to keep moving forward. And then your support, your help, your contributions are always appreciated. Thank you so much. So have a great day, night, morning. Thank you. Bye, everybody.
Thank you.
Bye to everyone.